Hi, my name is Melina Kibbe from Northwestern University. I was really happy to moderate a session that was called The Next Best Thing in Cardiovascular Sciences. This was really a lightning round session where representatives of each one of the American Heart Association councils presented on what the next best thing is in their topic area. Here's a sample of some of the exciting talks that we heard.
Hi, I'm Jenny Van Eyck from the Director of the Advanced Clinical Biosystems Research Institute at Cedars-Sinai Medical Center in Los Angeles. And I've been asked to talk a little bit about what is the next amazing thing that's coming down in the field of proteomics. So of course proteomics is the study of proteins and all their amazing diversity, both with respect to isoforms and their post-translational modifications. And really the reason we study proteins and be able to try to quantify them, either using immunoassays or things like mass spectrometry, is really that it is the proteome that drives the patient's phenotype. And the proteins that are within the cells and how they change with disease is really what drives the need to be able to have therapeutics and to be able to deliver the diagnostic markers that will help us to understand the process of the disease. So proteomics really is the phenotype, and being able to quantify it allows us to do that. So as a patient undergoes their progress in disease, we're able to capture all that diversity and be able to go in and look for both their past as well as their future and their immediate response to drug therapy. So proteomics has undergone a huge transition over the last 20 years in development of tools. And at this point, we're at this really very magical point in time where proteomics is able to quantify literally thousands of proteins and their diversity in really ability and in large numbers of samples never previously been able to do. This opens up both the ability to increase our understanding at the mechanistic or basic research level, but what's so exciting is we're now being able to also deliver some of that tools into the field of clinical application as well as population science. So for example, immunoassays are what we use in the clinical chemistry lab to be able to, for example, give you the cardiac troponin I assay that you use to be able to determine if someone's having an MI or having some kind of myocardial injury. So at this point, having higher sensitivity of troponin I is both a benefit and also a problem. New ELISA technology platforms are coming along that are very sensitive to be able to monitor cardiac troponin I. But what's interesting is we're able to also use mass spectrometry to look at modified forms to be able to give you really a risk prediction of whether or not someone coming into an MI, into emergency department with a positive troponin I actually will go on to have an MI within the next 24 or 48 hours, or whether or not it's just really an underlying cause that you'll see maybe 10, 20 years from now, an increase in cardiovascular risk. So these are developments coming from the area of proteomics and really using the tools like mass spectrometry to get these kind of unique biomarkers into the clinical assay. When you would, as a physician, then would ask for, let's say, a modified cardiac troponin assay, it may come either as an ELISA or as a mass spectrometry-based method. It should not matter. The data will look the same, will have the same quality, the same precision that's required. But this is really the field of the tool development that's coming along. Equally exciting is the area of population science, where we have dried blood spots, where you can go out and have really thousands of individuals have dried blood spots. Take them, prick just as you would if you had diabetes blood tests, dry them out, send them into a clinical lab, and we'll be able to give you really quantitative assays on proteins like CRP and BNP. Probably not cardiac troponin I yet, that's very high sensitivity assay, and we cannot yet do that on a dried blood spots. So tool development is very important in all types of area. And the newest area of technology is called SWAT, the data independent analysis, where really this technology allows us to do thousands of samples. And you can envision a time where we'll be able to do that on, fibroblasts that have been de-differentiated back to iPS, to cardiac myocytes, and we're treating all different types of drugs on them and finding really that drug that's specific for that person, or being able to, continuously monitor the blood of patients coming in and being able to develop new biomarkers as we go on. So precision medicine is really the proteome of the individual, and being able to monitor that and assess it by proteomics and the quantitative assays that we're able to develop is really the future and the excitement in our field. Thank you.